All right, thank you very much. Again, I'm Jane Lebkowski. I'm the president of Regenerative Patch Technologies, and I'm going to be telling you about the development of a bioengineered implant for the treatment of the dry form of age-related macular degeneration. And just a little bit about the company. The company was formed in 2012 to advance the development of technology that came out of a consortium of 3 universities and 1 academic center, the California Project to Cure Blindness from USC, UCSB, Caltech, and with manufacturing done at the City of Hope. The target indication really was AMD, in particular the advanced dry form of AMD. It's a very large market, I think, as we all know. And in developing this product, we leverage funding for both the preclinical phases, everything through the IND, and the execution of the phase 1/2A clinical trial that I'll be discussing through CIRM, the California Institute of Regenerative Medicine, who we are very grateful for to have actually helped support us in the development of this particular product. Right now, we have completed enrollment in our phase 1/2A clinical trial and have 1-year follow-up on that. We're preparing, we've been discussing with the FDA and have the protocol ready for our phase 2B clinical trial. And we have issued impending worldwide patents that cover essentially every aspect of the technology. So if we look at age-related macular degeneration, what is the exact problem? Well, it's an enormous problem. There are over 10 million people in the United States with dry AMD, and it's estimated that there are 196 million globally starting in 2020. And the incidence is actually increasing with the aging populations. Again, what is its manifestations? It's blindness. It's central blindness. It's looking at depriving people of independence through essentially robbing them of central vision, ability to read, ability to drive, and increased falls, and essentially increased dependence on caregivers. And unfortunately, to date, there are no approved therapies for this indication. So what is the pathology of the dry form of age-related macular degeneration, or geographic atrophy, which is the most advanced form? Essentially, what happens is, progressing through age, there's a degeneration of the retinal pigmented epithelial cells that happen in the retina. And these are the cells that are critically important for supporting the survival of photoreceptors and, in fact, the entire retina. They provide metabolic support, they provide a variety of structural support, and they allow for, in this case, the transportation of phagosomes and also other growth factors that are important for survival of photoreceptors in the retina. Another structure that is very damaged in the dry form of AMD, and including geographic atrophy, is the Bruch's membrane. And it's the Bruch's membrane that is the membrane upon which these RPE cells sit and polarize. And the cells have to be polarized in order to function. So our hypothesis at this point was to develop an implant that has RPE cells on the surface of this implant and supported by a scaffold. And that scaffold would, in fact, mimic that of the Bruch's membrane. So you have a mini tissue implant that can be used for the treatment of this disease. This is just a picture of the implant that was developed. Again, it's retinal pigmented epithelial cells are derived from human embryonic stem cells, and that they're plated on a engineered perylene membrane, and that perylene membrane is essentially used, and it has been engineered to mimic the diffusion properties of the Bruch's membrane. So on the right-hand side, you can see the actual membrane. It's about 3.5 by 6.5 millimeters in surface area, and it has a little handle, and it has what we call the nose, which is a landmark for the surgeon to determine which way to implant it, because this is polarized. There are cells on the top. So again, there are two very critical components for this particular implant. There are the RPE cells. Again, they're derived from pluripotent stem cells, specifically human embryonic stem cells, and that they're mature. We create vials of these cells that are cryopreserved. They do polarize appropriately according to the way RPE cells should function in the retina, and they produce a variety of different growth factors, particularly PEDF, that is important for the function of these cells. The engineered membrane came out of Y.C. Tai's lab at Caltech, and it is an ultra-thin parylene membrane. It essentially is 0.4 microns thick, and essentially it's an interesting membrane in that it can be folded so that without causing creases, or we can fold it up to form somewhat like a taco shell for delivery. We can engineer it to a very precise thickness, so it does in fact mimic the diffusion properties of the Brooks membrane, and that it's a flat surface. It's not porous, so that cells can't go through it. We've also engineered a delivery device, in this case a tool. We call it an insertion tool. This insertion tool has a forceps at the end that can grab the membrane and retract it into a shaft. And that shaft will allow it to, in fact, be delivered and protected while we're delivering it to the subretinal space. What the important thing about this particular tool and the folding of the parylene membrane, it really minimizes the retinotomy size. Our retinotomy is about 1 millimeter in length, and as a result can really manage to reduce the incidence of any kind of toxicity associated with making large retinotomies. This is just a video here. I don't know if you can play it in the back. That shows the delivery. In this particular case, essentially there is a vitrectomy that happens to remove the vitreous humor from the eye. There is in this case, a cannula is inserted to create a bleb in the subretinal space. And subsequently, you can see this is the tool, the insertion tool. As first, we're creating the retinotomy using a laser. Then we are taking the implant, removing a handle that we use for handling it. It is taken up by the insertion tool and curled like a taco to protect it during delivery. It is delivered through that particular retinotomy site into the subretinal space, and it is also then deployed. Subsequently, there is a tamponade that is applied in order to lay and secure the retina. So how do we manufacture this? For our phase 1/2A clinical trial, we manufactured this at the City of Hope. We created master and working cell banks of the embryonic stem cells, differentiated them, expanded them, created frozen banks of the RPE cells, and then under those circumstances took those frozen banks, vials from those frozen banks, and subsequently plated them onto the parylene membranes, and then subsequently have distributed them to the clinical trial site. So there's no animal-derived products used in production, and it has been successfully implemented in the clinical trial. We have manufactured now over 15 lots of this particular implant successfully and released them. The process is very, very, very amenable to automation. One of the achievements that we really focused on was to create now a cryopreserved implant, something that we could manufacture, cryopreserve, and distribute globally, and that is being implemented right now. So for the phase 1 clinical trial, we looked at looking at patients looking at the safety and potential activity of the implant in patients with advanced disease, advanced geographic atrophy. It was a single arm open label study. again, where the indication was advanced dry AMD with significant geographic atrophy. These were very, very significantly impaired patients. All patients were legally blind and being 2200 or worse visual acuity. And it was the worst eye that was treated. Some of the patients in the clinical study had only hand motion in terms of their visual capabilities. The implant is one size fits all. It's one implant. It has about 100,000 cells. And again, the primary endpoint was to look at testing the safety and tolerability of the implant and looking at a variety of visual function scores. The preliminary results were published in Science Translational Medicine in April of last year for the first 5 patients in the study. And it was executed by USC, 6 different sites at USC, at Retinal Vitreous Associates, Bob Avery in Santa Barbara, Praveen Dugal at RCA in Arizona, Clement Chan in Southern California, and Sanford Chen in Southern California. So what did we find out? Okay, first of all, we found out that the surgery could be completed. We could very successfully deliver the implant over the area of geographic atrophy, whether it was a person with a large area of geographic atrophy on the left-hand side or on a patient with a smaller area of geographic atrophy on the right-hand side. The implant was stable. We have followed these patients out now for over a year and find that the implant doesn't move, it doesn't, that there is no shifting, no wrinkling, nothing in terms of the actual movement of the particular implant. We've also been looking at a variety of efficacy measures, and first looking just structurally, what does this do to the retina? And one of the things we were very happy to find was that, in fact, we could see that in the area of geographic atrophy, which normally lacks a structure called the external limiting membrane, that when we put the implant over that area, we could see the recovery in some patients of the external limiting membrane. And importantly, we did not see disruption of the external limiting membrane when the implant went over an area that was not an area of geographic atrophy. We also have found now that some of the patients in the clinical trial have had improvements in fixation ability. Again, these are very, very severe patients. In one example here where a person had an improvement from 62% fixation to 99 to 100% improvement in fixation in approximately 120 days. We've also looked at visual acuity. This is data from the first 4 patients, and in this particular case, we've seen patients have either very stable visual acuity, not the normal decline that you might be seeing with patients who these normally progress. And we've seen in about 25% of the patients now actual improvements in visual acuity. And one patient here, in fact, had a 17-letter improvement in visual acuity. So we're going to be reporting all of the data from all 15 patients now that have been presented and have been followed up for a year in this particular trial, and that will be coming soon. So kind of in summary, you know, we've developed an implant to address the exact disease pathology that is found in the dry form of age-related macular degeneration. The process has been very stable right now. Manufacturing process is established, and we've manufactured multiple lots of the material. The phase 2A clinical trial shows very promising safety and activity profile in terms of efficacy and even visual acuity. And we're now moving on to our phase 2B clinical trial, which will really be establishing and testing this particular implant against endpoints that are registered, registration endpoints that are favored by the FDA. So we have key milestones going forward and an experienced team. So I'll just end there by saying that I want to thank all of the various people that were involved, from the teams at the universities, at the company itself, our clinical investigators, in particular CIRM and Santiam Pharmaceuticals, who have helped us support this program going forward. Thank you.